excellent talk. And yes, and it's our great pleasure to have Jonathan Michael to give today's talk. So John got his bachelor's from CMU and then did his PhD at Georgia Tech in, I believe, in Peter Younker's group, and worked on various different problems in sort of hierarchical and heterogeneous spatial organization of fibrous networks, and is very interested in computational modeling of both ex vivo and in vitro tissue organization and network organization. We in close connection with experiments, and now he's currently a postdoctoral fellow at Rochester Institute of Technology, working with Moumita, who's also over here in the audience. And today, I think he'll tell you about some recent work, which I'm quite fond of. I've heard Moumita speak about this on actually understanding some mechanics of heterogeneous tissues and having some interesting phase transitions in models of heterogeneous tissues. So take it away, Jonathan.
Okay, I want to first of all thank Suraj for that very kind introduction. I hope my talk lives up to that introduction, and I want to thank all the organizers today for the opportunity to talk with you. As Suraj mentioned, I'm currently a postdoctoral fellow at RIT, working with Moumita and collaborators, as was mentioned, I've been here about 2 years now. Before that, I was a graduate student at Georgia Tech. And over that time, I like to think I've now built up some expertise about the mechanics of filamentous media. With the guidance of some of my experimental collaborators, I've tried to get a little bit savvier about some of the biochemical particulars as they pertain to articular cartilage. So I guess what motivates us, well, broadly in the research group with which I'm collaborating, osteoarthritis. So osteoarthritis happens as the cartilage that cushions our joints degrades, and now this can happen either with gradual wear and tear over decades, or this can happen in response to acute injury. Now, once it does happen, in our later adulthood, we have limited ability to regenerate this tissue, and this has become a leading cause of disability and loss of quality of life and loss of productivity. And so it's desirable to understand how we might be able to arrest the progression, how we might be able to perhaps design savvier therapeutic interventions or better prostheses that more faithfully mimic native tissue to restore mobility and quality of life to people who are afflicted. So towards this aim, kind of an outstanding challenge is to develop a more mechanistic understanding of what goes wrong and why degradation or injury can lead to a change in the behavior of this tissue. And in particular today, I'm going to be exploring the shear mechanics of this tissue. The compressive behavior of this tissue has already been fairly well characterized using the successful model of poroelasticity, now worked out some 4 decades ago. But the shear mechanics of articular cartilage has received comparatively little attention. And so to kind of prepare us to delve in to the shear mechanics, I'd like to first take you through kind of a theorist's overview of the constituents of this tissue that are going to be salient for coming up with a mechanical model. So the cartilage in our joints has a scaffold that's made of collagen 2. And then this scaffold is embedded within a polyelectrolyte gel. This structure is very spatially heterogeneous. So you can see as depicted schematically in this diagram, the tissue is partitioned into essentially 3 regions: a shallow zone near the surface of the tissue, a mid zone, and a deep zone. So in the shallow zone, the cartilage scaffold is oriented more or less parallel to the surface, whereas towards the middle of the tissue, there's somewhat less orientational order, and then towards the deep zone, the scaffold is roughly perpendicular to the bottom surface, and moreover, the concentration is highly depth dependent. So generally speaking, this collagen scaffold becomes deeper and deeper, greater and greater the deeper you look into the tissue. And then, as I said, this this scaffold is ensconced within a hydrogel. So the components of this gel have as their backbone hyaluronic acid. So you might have heard of that if you've seen recent advertisements for beauty products. But then there are these beautiful bottle brush structures that branch off of this backbone. So there's kind of a backbone to this bottle brush. So as you can see, this is a very hierarchically organized structure. So these are referred to as proteoglycans, and then branching off from these proteoglycans, kind of shown in blue, are glycosaminoglycan or GAG molecules. So those are kind of the main players in the first narrative that I'll be relating to you today. How do we actually get our hands on one of these samples so that we can start to have something to say about its mechanics? Well, for this,
Jonathan,
please.
Can I quickly interrupt for a short question? How common, how generic or universal is this structural organization, especially with the orientation and the density gradient across different joints in a same animal and across all vertebrates? So how much variability is there in the structural organization?
Sure. So I guess what I can tell you is the experimental work that I'll be showing you today is carried out by harvesting explants from calf knees. It was an animal model that was readily available. I think they exhibit fairly similar structure to human cartilage. So I think among terrestrial vertebrates, among mammals, I think that one sees this motif occur fairly consistently. I'm not sure in other branches of the animal kingdom if the articular cartilage looks markedly different. Now, I know also there's the cartilage, for instance, in your jaw and your temporomandibular joint is a bit different from what I'm showing you for the cartilage in the knee. So there are some context dependent characteristics here that vary from one application to the next. Yeah. So strictly speaking, what I'm telling you is only validated for knee articular cartilage in mammals. So I couldn't comment upon necessarily lizards, but
thanks.
Yeah, of course. So let's see. So I guess I just wanted to mention, I've been fortunate to work with a skillful graduate student, Thomas Wise Jackson. All of the experimental work I'll be showing you in this part of the talk is principally the work of Thomas. And so I'm certainly indebted to him. So what Thomas has done is to take plugs out of the cartilage of recently sacrificed calves. Now these plugs are then coated with an epoxy resin, and then they can either be subjected to trypsin, which is an enzyme that digests away the proteoglycans in the polyelectrolyte gel surrounding the collagen matrix, or else kind of left as control healthy tissue.
Jonathan, sorry, yes. One more minor interruption. So Sonia actually had a question in the chat, I believe, about a figure on your previous slide. What are the percentages referred to in the figure from Gahunia?
Okay, let's see. Okay, so
are the percentages the percentage of collagen in each layer?
I think that this is rather referring to the approximate portion of kind of fraction of the tissue partitioned into each of these zones. Okay. Yeah, so I'll show you explicitly some experimental data in a couple of slides for the concentration dependence of, or for the depth dependence of collagen concentration. But this is just kind of a rough partitioning of shallow, middle, and deep. So I'll show you some more quantitative data in just a moment. To wit. Thomas has undertaken some Fourier transform infrared microscopy to measure the depth of both collagen and aggrecan as a function of depth. Now, I guess aggrecan is one of these aforementioned proteoglycans that branches off from the hyaluronic acid backbone in the hydrogel that encases the collagen scaffold. So we're going to use aggrecan here as kind of a proxy for characterizing how much the degraded tissue has been degraded by the addition of the trypsin enzyme. You can see in both the healthy and the degraded tissue, the concentration of collagen is substantially similar. So the trypsin largely spares the collagen, but then you can see in the shallow and the mid zones that the degraded tissue now has markedly less aggregant. So this hydrogel surrounding the collagen scaffold has now been greatly compromised. And so this gives us a model for studying a tissue that's been subjected to chemical degradation, and puts us in a position now to study how its shear mechanics will change in that situation. So then from there, the protocol is to fluorescently stain the tissue, and then one can photobleach a Cartesian grid into the tissue to track the deformation field. And then the tissue is subjected to kind of a low amplitude, low frequency shear to measure its complex shear modulus. When we do that, we find that whereas in the deep zone, the shear modulus is similar for healthy and degraded tissue, in the shallower portion of the tissue where this hydrogel has been kind of eroded away, we see a significant difference between the healthy and the degraded shear modulus. So now the shallow and mid-zone portions of this degraded tissue have a much lower ability to bear an applied load. And so the challenge to us as the computational modelers was whether we could make sense of this in terms of the experimental data that we had at hand. So to begin,
Jonathan,
oh yeah, please.
The G star that you have, the shear modulus, at what frequency was this measured? Because this is oscillatory rheology.
Yes, very good. So that was at 1 hertz.
Okay.
Yeah, okay. I didn't prepare a lot of material about this, but our experimental colleagues looked at the real and complex components and found that while there was a non-zero out of phase loss modulus, it was orders of magnitude smaller than the storage modulus. So so you're looking at just the 1 hertz amplitude of the shear modulus here.
And presumably, and what are the colors? I sorry, I might have missed this. What's blue and what's red?
No, I think a legend got clipped off here, so that's my mistake. Red here, pardon me, is the degraded tissue, whereas blue is the healthy tissue. So pardon me, I think that was my oversight. Of course. Okay, let's see. All right, so
there's one more question in the chat.
Sure.
Andrei Zakharov, the measured shear modulus is much larger than for collagen networks. Why is it much stiffer?
Okay, so the gel itself actually has a pretty large shear modulus, on the order of a couple of 100 kilopascals. So this, as I mentioned, this gel is a polyelectrolyte, and so the previous modeling, for instance, using a Poisson-Boltzmann approach has shown that the mobile charges within this material contribute immensely to the overall shear response of the tissue. Like if you bathe this polyelectrolyte in a bath of counter ions, you can lower its shear modulus by 200-300 kilopascals. So it's true that this is much higher than what you would see if you just had a bare collagen network. Okay. So let's see. So I guess in putting together our model, we start by kind of looking at this full picture where we have a collagen kind of shown in these thick striated bands immersed in this gel, and to get started, we wanted to just focus on the collagen scaffold. And when we look at this as computational modelers, we're reminded of a now well-established framework, namely a rigidity percolation theory. So we are going to abstract away a number of the biochemical details for just a moment and idealize the collagen scaffold as a lattice of spring-like bonds. So these bonds can resist both bending as well as stretching and compression. And by varying the density of bonds that we have in this network, we can tune the stiffness of the network until eventually the network kind of exhibits a dramatic drop in shear modulus. It's kind of reminiscent of a thermodynamic phase transition. So to wit, here I'm showing you a Kagome lattice with a nominally a valence of 4, but here with just 40% of bonds, a slightly denser network with 60% of binds retained, and a denser network still with about 90% of binds retained. So we kind of liken this process of varying the amount of material we retain in this model network to varying the concentration of collagen in the articular cartilage. So we take in our model, we'll take the collagen concentration to be in correspondence with the fraction of binds that we retain in our network. And so in these images, I've just kind of paired away material in a completely random, spatially homogeneous fashion. So then, I guess to make this idea a little bit more precise, I mentioned that this network could resist stretching. So here we ascribe these binds a spring stiffness alpha, so that we then penalize the stretching, where here u sub ij is the relative displacement of two vertices in the network I and j, and then r hat is the unit vector pointing from vertex I to vertex j. So this is just a harmonic penalty essentially on stretching. And here the sum runs over all pairs of adjacent vertices I and j connected by a bind, where p sub ij is 1 if the bind is present, and 0 if the bind is absent. Moreover, as I mentioned, we also penalize stretching according to bending as according to a bending modulus kappa. So here we now have triples of vertices because bending involves two initially collinear adjacent bonds. And so if the bonds between I and j and k are all present, then given triple, we'll also have a bending interaction. So with just these ingredients so far, if I subject one of these model networks to a small shear, well, if I only retain 50% of the binds, essentially the network is completely, can respond to that applied shear without any of the individual binds having to bend or stretch or compress. And so this the strain that any particular bond takes up in this network is negligibly small, and this small shear deformation will cost me negligibly little energy. Now, if I increase the fraction of material I keep just a little bit, now there's no longer a zero energy way to accommodate a shear deformation, and so we see now that there's this percolating network, hence the name, of bonds that are under strain. And so now it's costing some non-negligible amount of mechanical work to deform this network.
Hey, Jonathan, can I ask a quick question?
Yeah.
What is the reason for choosing a Kagome lattice over some of the other common lattices like triangular? Does it have to do with the coordination number being 4 and that being preferable?
Right, that's just it. So our idea was that the vertices in this network tend to the vertices in this network correspond with places in which adjacent fibers in the collagen scaffold and the tissue are cross-linked chemically to one another. And to our knowledge, the Kagome lattice gives a more appropriate distribution of valences than some of the more highly coordinated alternatives.
So there is a follow-up question related to that. I'll just read it from Andre. He's asking, what is the coordination number that you get, average coordination number after dilution?
Sure. So for the fully coordinated network, so then one can actually just multiply that full coordination number by the portion of binds retained. So here, I suppose 2.2 for instance, would be the mean coordination number for this structure.
And can I ask a question about so if I understood the experimental data that you showed first, that was measuring shear response in the presence of compressive load. Is that correct?
Yes. So, when we stand on our joints, of course, we're putting them under a little bit of compression. And so yes, the experimental apparatus kind of mimics that by applying a slight compression to the samples, and we've mimicked that in our simulation protocol. So here, we're considering, and once again, kind of a low amplitude response. So I'm not seeing strong evidence of strain stiffening in this regime. I guess there's some debate about the role of strain stiffening, just because if this tissue is strained too highly, then that means that other tissues responsible for stabilizing the joint and preventing overly large translations of the joint have failed, and the tissue and the joint is at risk of catastrophic injury. So we're definitely also interested in the way that tissues in the body could ultimately fail, but yeah, I don't won't be touching upon that much today.
Okay. And as of now, the network, so Kagome, is entirely isotropic, and your dilution, I'm guessing, is also isotropic. Do you all also do anisotropic versions of rigidity calculation?
Yes. So actually, kind of the second story that I'd like to tell today.
I'll wait for it. Thank you.
Yeah, but excellent setup for the second part of my talk. So thank you. Okay. Let's see. So if there's nothing further at the moment, so I guess now I've shown you this stripped down picture of the college and scaffold, but of course the devil is in the details. So now if we bring back the gel, we add a third term to our model, apart from the stretching and bending, and that's a term describing a coupling between the network and the gel. So here we introduce a parameter mu that corresponds with the shear modulus of the gel. So we then penalize relative displacements of adjacent vertices transverse to the vector separating them. So the idea is that that transverse component of the displacement corresponds to those two vertices applying a shear traction on the gel, on the volume of gel that separates them. And so this final term captures the network gel interaction. So with these ingredients in hand, we then introduce a simulation protocol in which we pin the bottom vertices of the network. We apply periodic boundary conditions at the left and the right. And then we apply a modest few percent compression, and then shear the network to the right. So we'll consider 5 different shear amplitudes for a fixed compression. With the constraints that I mentioned, we then relax all of the other degrees of freedom in the network. So all of the vertices apart from the top and the bottom vertices. And we relax into the kind of the mechanical ground state of the network and compute the residual strain energy. And so then by tabulating the strain energy at a few different strains, we can numerically differentiate that energy with respect to the amplitude of the applied shear strain to pull out a differential shear modulus, which will then afford comparison between our model and the experimental data. Okay, now the model that I've presented to you so far might seem a little phenomenological, but we have actually had a lot of success in making contact with the experiment. So I want to just briefly walk you through how we go about that. So G is our overall kind of experimental measured shear modulus. So then we introduce kind of a scaling constant C that takes us from simulated units to experimental units. We then have a background modulus. So this corresponds just the contribution due to the fact that there's a gel that's having to shear as well as a network. So mu naught is kind of the floor that we see in the absence of the constituents that are degraded by the trypsin enzyme. So we can estimate mu naughts from data in the literature about the concentration and molecular weight of hyaluronic acid, which is the principal component of the gel that's left after degradation. And we find that our model fit is in good agreement with the predictions that we make using the theory of elasticity for hydrogels. We also consider that this gel can be stiffened by the presence of aggrecan, whose density is indicated by rho A, and collagen, whose concentration is here indicated by rho C. Beta and gamma are determined by fitting, but can be constrained once again by experimental knowledge of the shear modulus of this hydrogel obtained from the literature. And then finally, we have a G network obtained from our network simulations, where delta times the collagen, delta is a fitting parameter, delta times the collagen concentration is a mapping from collagen concentration to a bond portion, which is our simulation parameter. Kappa is that simulated bending rigidity of an individual fiber. And then the final term is just that background shear modulus that I discussed a moment ago. So while we do have a few fitting parameters, we have a large amount of data to work with, and we have a lot of constraints that we can apply on these fitting parameters based upon physiologically relevant ranges that we've inferred from the literature. So when we do this, we find we can very successfully pass a surface through both the healthy data that are shown in blue and the degraded data, which are shown in red. So along one axis, we see the aggrecan concentration. So that's one of these so-called proteoglycans that's branching off of the hyaluronic acid backbone of the gel. Then we see the collagen concentration, which we note was very similar between the healthy and the degraded tissues. And then in the vertical axis, we're seeing once again the amplitude of the shear modulus measured at 1 hertz. A striking thing that you can see here is that a rough doubling of the collagen concentration results in over a decade increase in the shear response of the tissue. So this model not only handles a wide range of parameters in with regard to both proteoglycan and collagen concentration, but also spans a couple of decades in the shear response of the tissue. So with this model in hand, what we envision is that if a patient, say, presents with a joint injury, complains of certain symptoms, then perhaps a non-invasive procedure like an MRI could be used to image the tissue, infer different concentrations of different constituents. And then with a constitutive model like this, one could infer how the chemical and structural changes observed using some non-invasive imaging technique might lead to the symptoms that the patient is experiencing, and then this could be used to inform a more strategic course of treatment. Okay, and I'm being
Jonathan.
asked by Dan Beller. Yes.
Oh yeah. So Dan asked a question in the chat, which is also a question that I had, which is what represents aggrecan in the model?
Yes, very good. So maybe I'll just walk back here. So kind of going back to this energy accounting. So mu, is our model parameter representing the shear modulus of the tissue. And so then we have kind of a simple scaling model for what this should be in terms of the concentration rho A of aggrecan, which we're using as a little bit of a proxy for the other constituents of the gel, and the concentration rho C of collagen. And I guess we picked the simplest model we could write down just to get going, but it makes predictions that are in good accord with published values for the shear modulus of this hydrogel. Now, so I guess broadly, aggrecan is kind of a proxy for the constituents of this background gel, apart from the hyaluronic acid, and so it's going to kind of control the shear response of this gel, and that can be modulated by a couple of hundred kilopascals, just due to the variation in the background gel. Okay, did that lend some clarity? I can.
Yeah, I think so.
Take another step at that.
No, I think it's okay. Go ahead. Yes.
Okay. Yeah. So I just had one other story that I wanted to tell today, and I just wanted to make sure that I was budgeting my time here. So I guess the big takeaway from this phase of the talk was that we've now have kind of a proof of principle for this mechanistic model relating concentrations of individual constituents to the overall bulk properties of this tissue, and this lends greater understanding into how chemical degradation of this tissue can result in loss of function. Now I'd like to pivot a little bit. Suraj asked an interesting question several minutes ago, namely, have we thought about what happens if we have now an inhomogeneous dilution of the bonds in our network? And so we were inspired to think about just that very thing by images such as these. So what I'm showing you here are some kind of model tissue sections. So these were made by implanting cells called chondrocytes, which are responsible for generating the collagen matrix inside of cartilage into a hydrogel scaffold. this was then implanted in a mouse model and harvested at varying times, after varying amounts of times had elapsed. And so what one can see stained in red are proteoglycans, once again in this kind of background gel that encompasses the collagen scaffold. And what you can see is that this structure doesn't grow in a spatially uniform way. Rather, it seems like they're kind of nucleation sites, and that the tissue kind of fills in as it grows out from these nucleation sites. And so then our group became interested in understanding what the consequence could be of now having spatially modulated density. So to answer that question, I'm going to have to update the mathematical model that I showed you previously. So before, as was discussed, bonds were plucked away in a purely random, homogeneous way. So now we take inspiration from recent literature modeling the formation of colloidal gels. So when we consider a bond as a candidate for inclusion in our diluted network, as shown in red in this lower image, we first look to its neighbors, and we consider two bonds to be neighbors if they share a vertex. So for the Kagome lattice in particular, this bond in the fully connected lattice has 6 nearest neighbors. One can show in general that this will be twice the coordination number of the lattice minus 2. So then we choose a correlation strength, C, and we include the candidate with a probability 1 minus C to the power of 6 minus the number of nearest neighbors. So for instance, if C were 0, then this probability of inclusion would be 1. And so this would reduce to the case of completely random homogeneous dilution. And then the larger that C becomes, the more spatial modulation you see in the concentration of bonds, and you start to see kind of dense, patchy structures separated by sparse, spindly regions. So I'm kind of showing you a slice of that parameter space here. So moving up in this figure, kind of see the effect of increasing the correlation strength for a given amount of bonds retained. Then moving to the right in this figure, you see the effect of including more and more material. So you can see moving upward, you see the network kind of segregate into these dense clusters tethered to one another by slender bridges. So now I'd like to ask, what's the consequence of this kind of spatial modulation in the for the shear mechanics? Well, we use the same kind of shear mechanics simulation protocol described previously, and so if we look at the shear modulus in simulation units versus the portion of bonds retained, we see that initially, as we increase the correlation strength, the network has a non-negligible shear modulus for a progressively smaller portion of bonds retained. But interestingly, this effect appears to saturate above an optimal value of about C equals 0.6. So you can see in the final brown curve corresponding to a correlation strength of 0.8, the network actually becomes rigid for the first time for a larger portion of bonds than is required when we have a correlation strength of 0.6. So kind of looking at this from a bird's eye perspective, we can see that there's this contour of incipient rigidity in this correlation strength bond portion space exhibiting it's what we would call a reentrant dependence of threshold fraction of bonds needed for a rigid network. And if we focus in particular on this transition region, we find that we can consistently observe power law-like scaling of the shear modulus with the excess bond portion. So that is the actual portion of bonds minus the critical portion, PC, which is the kind of the minimum amount needed to form a marginally rigid structure. So here we denote by PC the critical bond portion, and by beta kind of the exponent with which the shear modulus scales with excess bond portion. Now, using this quantitative model, we once again kind of recapitulate the behavior that we picked out by inspection, showing the scaling of the critical bond portion versus the correlation strength. But interestingly, the scaling exponent exhibits the opposite behavior. So we initially see that the shear modulus increases more abruptly with an increasing fraction of bonds retained. And then past this optimal correlation strength of 0.6, we see that once again, the scaling exponent decreases. Broadly put, the larger the critical bond portion, the lower the scaling exponent. So it would seem as though somehow this correlation strength of 0.6 is offering us an optimal economy of material. You're getting the most bang for your buck. And so we'd now like to kind of turn to a micromechanical assessment of the deformation mechanics of our networks to get some more mechanistic insight into why this might be the case. So to begin with, I'd like to start, I'd like to tell you about the idea of non-affinity. And okay, I imagine some specialists in the audience will be acquainted already, but essentially for a simple elastic continuum subjected to simple shear, as in our current simulation protocol, you would expect that a simple linear transformation would describe the displacement of any point inside the material. And this assumption actually often holds good for a lot of rubbers, for instance. But for stiff biopolymer networks with a large persistence length in comparison to the mesh size of the network, this is often found to be violated. So then we introduce the non-affine displacement as the true displacement of each vertex in this network minus the ideal affine displacement given by this simple linear transformation you see here, in which the x displacement is proportional to the height of the vertex times the amplitude of the shear strain. So this quantity of non-affinity has been widely attested as a means of quantitatively describing the mechanical anisotropy or heterogeneity of a network. So then if we look at the 2-point correlation in this quantity azimuthally averaged, we see that this quantity tends to decay exponentially with the separation between a pair of points. Now I'm showing you a representative set of curves here for a correlation strength of 0.6, but we saw qualitatively similar behavior for correlation strengths ranging from 0 to 0.8. So from the exponential decay length in this curve, we can identify kind of a characteristic mechanical length scale, if you like. This correlation length scale follows a qualitatively similar pattern for different correlation strengths, but you can see that at low fractions of bonds retained, the correlation strength is highly stratified according to how correlated the network is. So some more highly correlated networks have much more gradual decay in their non-affine displacement correlations. So we can go through and pick out the correlation length at kind of the critical bond portion for each correlation strength. And so here on the y-axis, I'm showing you once again this non-affine correlation length, but specifically at the point of rigidity percolation. This appears to diverge with correlation strength. I'm currently running additional simulations to see if I can get another decade here, but we already found this result to be quite striking. And what I thought was interesting was that then if I plot the critical bond portion versus the critical non-affine correlation length, we see the same kind of reentrant scaling that we saw when we plotted the critical bond portion versus the correlation strength. So the picture that's emerging, at least in my mind, is that as we introduce a little bit of structural correlation into these networks, we create these dense, rigid clusters, and coupling of these dense, rigid clusters enables system-spanning force chains to emerge, and this enables one to make a rigid network with less material. Whereas if these rigid clusters become too large, then they become kind of poorly mechanically coupled to one another. So you have these kind of large chunks of material, but scattered few and far between throughout the network. And so at some point they become too poorly mechanically coupled to one another to allow the network to efficiently transmit stress. And so you see that beyond this correlation strength of about 0.6, you get too much of a good thing, and having a larger kind of non-affine correlation length scale actually begins to do more harm than good. Now, as one final point, I wanted to talk about kind of a preliminary matter that I'd identified here. So if we go back and look at this network that's just above kind of the threshold for marginal rigidity, we see that there's a huge variability in the amount of strain that bonds in the network take up. So some of the bonds in the network are much more highly strained than others. So as a kind of a measure of the statistical dispersion, I consider in particular the skewness in the log base 10 of the strain. So here the skewness is the third moment about the mean, normalized by the cube of the standard deviation. So just to kind of give you an idea of what a couple of these distributions might look like for a couple of representative cases. Here I'm showing you the distributions for two networks, both with a bond portion of 0.6, one with a correlation strength of 0 and the other with a correlation strength of 0.8. So you can see in one case, there is a comparatively tight distribution of strain magnitudes for 0 correlation strength. But then when we consider the case of C equals 0.8, we can see that there's this high peak towards the high end of the range of strains, but then there's a longer tail, and there's kind of a larger subpopulation at lower values of strain, and so as these distributions become more skew, we kind of hypothesize that it becomes more and more likely that certain of the bonds in the network will become overstrained and snap, leading to crack nucleation. And so, apart from considering the economy of material for low amplitude deformations, it might also be relevant to consider the liability that these materials will eventually fail. And so there are more trade-offs to consider than merely thinking about the shear response for low amplitude deformations, and these might be relevant both in practical applications, if one is trying to design a novel class of hydrogel, or in trying to understand the susceptibility of tissue to injury. So I know that I'm nearing the end of my time here. So just in concluding, of course, I'd be remiss if I didn't note that I'm part of a highly collaborative interdisciplinary research team, and I'm deeply indebted to numerous advisors and colleagues. So I want to thank my postdoctoral supervisor, Moumita, as well as professors Lawrence Benasser and Itai Cohen at Cornell, and Thomas, once again, a skillful graduate student who furnished me with all of the experimental data that I showed you today. And I want to thank the DAS lab more broadly for their support and camaraderie over a trying couple of years for all of us. So in concluding, I want to note, first of all, that a strikingly simple model. At the March meeting, I even heard it called a disturbingly simple model. Nonetheless, offers striking predictive power in relating the shear response of cartilage to the concentrations of its individual constituents. Furthermore, spatial fluctuations in the density of biopolymer networks can have significant consequences for their shear mechanics, and some of the lessons I've learned here can inform us about how to kind of thread the needle between economy of material in designing hydrogels or polymer scaffolds, and avoiding potential hazards. And so, with that, I'd like to once again thank everyone for your kind attention, and the organizers once again for the wonderful opportunity to speak to you today. And if there are any further questions, I'm happy to entertain those now.
Thanks, Jonathan. Wonderful talk. So I'll clap for you, and I'm sure there's many virtual claps by the others in the audience. Okay, so questions from the audience, feel free to unmute and directly ask. And just to also note, I think after about 15 minutes or so, we'll all have the session with just graduate students. So graduate students, please stay online, and so you can participate in that session. So anyway, go ahead. Any questions from the audience?
Yeah, so I can start. So I suppose you're always below the Maxwell threshold, which is 4 in 2 dimensions in your simulations. And so I expect the shear modulus is kind of dominated by the bending, and so by the bending modulus. So is that something you can vary in experiments, such as by varying collagen cross-linking? Like, can the bending modulus of the collagen fibers be varied? And have you looked at the effect of changing the bending modulus parameter in your simulations?
So we've definitely looked at varying the bending modulus in fitting, simulation data to experiments. Experimentally, because that's an interesting suggestion. Yeah. So if we, I don't know of an easy way. This might be my naivete as a modeler to independently tune the bending modulus of the fibers without concurrently modifying the effective stretching stiffness. Yeah, I would definitely agree with you that the bending modulus is a substantial making a substantial contribution here, since yes, as you say, we're well below isostaticity. I know there's substantial capabilities, for instance, in our collaborator Larry Benasser's lab to print using various biopolymers, and so perhaps they could cook something up. I don't have any results on that just yet. I've just kind of played around in the simulation parameter space too so far. But yeah, definitely a provocative question and an interesting avenue.
Dan, go ahead.
Yeah, I was wondering if the second half of your talk relates to the idea of crack propagation or failure modes, if the connectivity is changing during the simulation.
Yeah, so I guess I'm not considering dynamic connectivity once we lay down the network. At least in the simulations I've showed you today, it has the same coordination number throughout. I guess I know, for instance, highly over cross-linked collagen scaffolds in bone in older patients have been related to a greater susceptibility to fracture, because that takes away some of the lower energy, less destructive modes of deformation, and kind of makes the tissue less pliable. And so I think that the notion of having too many highly coordinated regions as opposed to having some soft pliable regions could definitely have some implications for how susceptible the material is to fracture. I'm just now kind of getting into the fracture properties of these materials as a follow-up, so I don't have any hard results on that yet. I can just kind of speculate for now. But
thank you.
Yeah.
Okay,